What if I told you we could see the future? Not in some crystal ball, but inside something as tiny and simple as a seed. A future where our food is safer and more secure, all because we can finally see a threat that's been totally invisible. Until now.
I mean, think about that for a second. What if we could actually stop a plant disease, not after it's already torn through a field, but way before the very first root even breaks the soil? So why does this matter so much? Well, it really boils down to this one number, 90%. About 90% of the world's most important food crops, yep, the foundation of pretty much everything we eat, start their life as a seed. The health of these little guys is absolutely crucial for agriculture all over the globe. But there's a problem, a really big problem. There's this hidden danger lurking inside the very thing that's supposed to give us life, an invisible threat that can have, well, some pretty devastating consequences. Yeah, we're talking about microscopic stowaways, things like fungi, bacteria, and viruses. These pathogens can travel silently across the world inside a seed, and then, when the conditions are just right, they unleash diseases that can wipe out an entire harvest. With our world population heading towards 10 billion by 2050, those are losses we just can't afford. For decades, the ways we've tried to find these pathogens have been, well, a bit of a compromise. Sure, we could look at them, but that only catches the really obvious problems. Or we could use powerful lab techniques like PCR, but that means you have to destroy the seed to test it. And on top of that, they're often slow, expensive, and you definitely can't use them on every single seed.
But what if we didn't have to make that choice? What if we could give every single seed a non-destructive super scan that could see the chemical signs of disease with unbelievable accuracy? Okay, so this is where a technology called Hyperspectral Imaging, or HSI, comes into the picture. The easiest way to think about it is like giving a camera Superman vision. You know how our eyes see three basic colors: red, green, and blue. Well, an HSI camera sees literally hundreds of different bands of light, most of which are completely invisible to us, stretching all the way from ultraviolet to near infrared.
And here's the absolute magic of it. HSI can spot the teeny tiny chemical changes a pathogen causes inside a seed before any physical symptoms show up. It can find the disease when the seed still looks perfectly healthy to the naked eye, and, this is the best part, it does it without harming the seed at all. Now, I know, it sounds like something straight out of science fiction, right? But it's a very real, very systematic process. So, how exactly does this super scan work? Let's walk through it. You see, this isn't just guesswork. Researchers have laid out this clear 7-step workflow to create and double-check these detection models. It's a super rigorous method for training an artificial intelligence to become, basically, an expert seed pathologist. All right, let's break this down. First, the HSI camera scans a bunch of seeds, both healthy and infected ones, and captures an image. This creates what's called a data cube. Think of it as a detailed 3D map of each seed's unique light signature. After scientists confirm which seeds are actually infected using the old-school methods, all of that data gets fed into an AI. The AI then learns to spot the hidden patterns of disease. And finally, you test it on a whole new batch of seeds to prove it really works. So every single seed, when it's scanned, reveals this unique spectral fingerprint. A healthy seed has one kind of fingerprint, but a seed infected with a fungus or a virus, it has a slightly different one because of all the chemical changes going on inside. And the AI gets incredibly good at telling the difference, at spotting those telltale signatures of disease. Okay, so that all sounds amazing in theory, but does this technology actually hold up in the real world? Let's take a look at the results from over 20 different scientific studies. This data gives us a fantastic snapshot of the research so far, and one thing that really jumps out is that over half of the studies used naturally infected seeds. Now that's a huge deal. It means the technology is being tested against the real-world problems that farmers actually face, not just some infection cooked up in a perfect lab. It makes the results way more meaningful. And the results themselves? Wow, they are nothing short of remarkable. For Fusarium fungus on wheat, researchers hit 100% classification accuracy. That means stopping a blight that can make entire harvests toxic. For Aspergillus on corn, again, 100%, preventing a mold that's a serious health risk. Even for really tough-to-spot viruses in watermelon seeds, they're hitting accuracy rates above 90%. This isn't just promising. It's a total game changer. So, with these incredible results, you have to ask, what's next? What does the path forward look like for this technology to actually make our food supply safer? Now, it's not a perfectly clear path just yet. There are a couple of practical hurdles. For one, the HSI equipment is still pretty expensive, which puts it out of reach for a lot of labs and seed producers. And to make it all work, you need serious collaboration. You need plant pathologists, physicists, software engineers, all working together. That's a rare mix of expertise. But as the technology gets cheaper and more common, the potential is just enormous. I mean, imagine seed certification programs that can scan 100% of a seed lot, not just a tiny random sample. This could give farmers a level of confidence we've never seen before in what they're planting. It could dramatically strengthen the integrity of our entire global food supply chain, from that first seed all the way to the food on our plates. Which, brings us right back to where we started. This incredible new ability to see the invisible, to find a microscopic threat hidden inside one tiny seed. Could this be one of the most important keys to sustainably feeding our very visible and very hungry world?